So hello and welcome to the Introduction to DRAGEN on the NextSeq 1000 and 2000 webinar. My name is Ifeanyi Yesina and I'll be your presenter for today. There are going to be a lot of topics covered today, so let's get started. First, I would like to preface that this is a repeat of the previous NextSeq 1000 and 2000 Introduction to DRAGEN webinar, given in October of last year. However, this webinar has been updated to include many of the new features that have been introduced to the NextSeq 2000 DRAGEN analysis since then. We're going to break this webinar into two sections. During the first portion, we are going to talk about what DRAGEN is, what it does, and how to set up a run for analysis. This includes discussions about what is DRAGEN, DRAGEN compression and decompression, run setup options, DRAGEN on the NextSeq 1000 and 2000 versus DRAGEN on a standalone server, V2 sample sheets, and requeuing analysis. The second section of this webinar will focus on what to expect once the DRAGEN analysis completes. This section will include discussions on the following topics of where to find DRAGEN output files, the NextSeq 1000 and 2000 analysis workflows, specifically our new workflows, DRAGEN single cell RNA and DRAGEN Amplicon, how to update your DRAGEN workflow, switching between the different DRAGEN software versions, the DRAGEN reference genomes available, and the DRAGEN output files transferred by Universal Copy Service. So DRAGEN stands for Dynamic Read Analysis for Genomics. And of course, our focus is on the NextSeq 2000 DRAGEN, which I'll refer to from now on as the NextSeq 2K. DRAGEN produces common analysis files such as FASTQs, BAMs or CRAMs, and VCF files, and alignment and variant calling metrics. DRAGEN on the NextSeq 2K can be set up to automatically launch on the instrument or in the cloud after a sequencing run completes. So what is DRAGEN? DRAGEN is software. DRAGEN contains several workflows on the NextSeq 2K, including all of the workflows here. And DRAGEN is also hardware. DRAGEN uses field programmable gate arrays, or FPGA cards, and these FPGA cards are also known as the DRAGEN chip or DRAGEN card. The DRAGEN chip is programmed to execute computing tasks such as alignment, variant calling, and counting for DRAGEN RNA pipeline. The DRAGEN chip has also been engineered to accelerate the analysis. Together, the optimized DRAGEN software pipeline and the DRAGEN chip make up DRAGEN. Now that we know what DRAGEN is, let's talk about the ability to use DRAGEN's compression and decompression software. GZIP compression, or .gz files, is the compression format that Illumina has traditionally used for FASTQ file compression. The DRAGEN compression software will produce FASTQ.aura files, which reduces the data footprint of FASTQ files by a factor of 5 compared to GZIP FASTQ files. Both DRAGEN and GZIP compression are lossless compression formats. Lossless compression meaning that there is no data in the original file that is lost as a result of the compression, and the integrity of the data is preserved. How DRAGEN compression works is by rapidly mapping reads onto a reference and using a custom binary format to encode the reads as a list of positions and differences without losing any of the data in the original file. The reference used for DRAGEN compression is human reference. Using a custom reference is currently not supported at this time. DRAGEN compression is a great tool because it will generate smaller FASTQ files and reduce data storage costs and help with faster FASTQ file transfer. You can choose your compression format when setting up a NextSeq 2K run using BSSH Instrument Run Setup. Please note that DRAGEN FASTQ compression can only be performed locally on the instrument and requires DRAGEN 3.7.4 and above. The DRAGEN compression also requires a license to be installed, which is included at the cost of the instrument and no additional purchase is necessary. In this version 2 sample sheet, next to the FASTQ compression format field, you will see either DRAGEN or GZIP listed for the software to use. So DRAGEN compression will create the FASTQ files with an original read archive or .aura extension, and the DRAGEN decompression software can be used to decompress one file at a time back to FASTQ.gz format. The DRAGEN decompression software is also free for everyone to use, and there is no license required. This software must be run on the command line, and the DRAGEN decompression software can be downloaded from the Illumina support site. Okay, so now that we know how to compress and decompress FASTQ files, let's get into the run setup options for the NextSeq 2K. There are 4 different run setup modes. First, we have standalone mode. And for standalone mode, no DRAGEN analysis occurs on the instrument or in the cloud. Next, we have the local and hybrid modes. In both cases, DRAGEN analysis occurs on the instrument. These two differ in where the run is set up, either using a sample sheet for local only or setting up the run in BSSH using Instrument Run Setup for hybrid mode. Regardless of where the run is set up, the analysis will occur on the instrument when you're using local or hybrid mode. Lastly, we have cloud mode only. For cloud mode, DRAGEN analysis occurs on the cloud and the run setup uses Instrument Run Setup in the cloud on BSSH. Now that we are aware of the different run setup modes, I want to briefly talk about DRAGEN analysis on the NextSeq 2K versus DRAGEN on a standalone server. When analysis is occurring locally on the instrument or onboard the NextSeq 2K, DRAGEN demultiplexing will start after the index cycle base calls have completed sequencing. Preliminary alignment of read1 sample data to the reference genome will then start after the demultiplexing completes. This means that the analysis will start before the run fully complete on the instrument. In cloud-only mode, when the run is being analyzed in BSSH, demultiplexing and secondary analysis do not start until the run has completed and all the primary data has been fully uploaded to the cloud. Cloud mode does not mean that secondary analysis will be performed automatically. Now, if using standalone mode on the DRAGEN server for analysis, meaning that you're not using the instrument, demultiplexing and secondary analysis can be started manually after a run completes and all the primary data has been transferred to the standalone DRAGEN server. Okay, now that we are aware of the run modes, let's talk about how to generate V2 sample sheets for DRAGEN FASTQ generation. We highly recommend using the Instrument Run Setup in BSSH to generate V2 sample sheets. To access the BSSH Instrument Run Setup, once on the dashboard, you will click Run and then New Run and select Instrument Run Setup from the drop-down menu. Next, you'll be taken to the Run Setup page. First thing to do will be to select the instrument platform and run name to use. Next, it is important that the analysis location selected should be local when planning to use either local or hybrid mode, and BSSH if you are planning to use the cloud. If BSSH is selected instead, but the intention was to do local analysis, then this can cause sample sheet to fail validation when it is import onto the NextSeq 2K control software. Next, after selecting the analysis location, you will then pick the type of a DRAGEN analysis pipeline to run. Please note that you want to make sure that you are selecting the correct pipeline and version that is installed and running on the instrument. If you are unsure of what version you used, then you can get to the DRAGEN version by going to the DRAGEN settings within the NextSeq 2K control software to confirm the installed DRAGEN pipelines that are currently on the instrument. After filling out all the required fields and putting the rest of the sample and run information together, for local analysis, you can then export the sample sheet by going to Planned Runs and then exporting the sample sheet by clicking on the run, then clicking on the checkbox next to the run and selecting File, then selecting Download, and then selecting Sample Sheet to export the V2 sample sheet. You can then take it to the instrument where you're planning to run locally. This is what an exported V2 sample sheet will look like. All this information is pre-populated from the setup that was done in Instrument Run Setup. Please note that Illumina Experiment Manager, also known as IEM, which is used to create V1 sample sheets, will not be updated to generate V2 sample sheets. And when using the NextSeq 2K, we always recommend using Instrument Run Setup to create properly formatted V2 sample sheets that will look like this. So now that we understand how V2 sample sheets are created and how they drive local analysis, let's talk about kicking off local analysis by requeuing on the instrument. Local analysis can be requeued via the control software's process management screen. To requeue, the system state must be idle. meaning that no sequencing or DRAGEN analysis can be in progress. And the temporary run folder found in user local Illumina runs must not have been deleted or moved off of the instrument. The analysis cannot be requeued if the temporary run folder has been deleted, even if the user tries to move a copy back to the temp run directory. And lastly, the sequencing run may need to be completed successfully in order to requeue. To requeue, you would find the process management screen in the top right-hand corner. Click requeue, and note that the analysis runs that are errored or in sequencing where it is stopped, as shown on the screen, cannot be requeued. In this example, the runs below have an errored or stopped status, so the requeue option is faded or grayed out. When requeuing an analysis, a new V2 sample sheet may be provided and the output directory can be changed. To use a new V2 sample sheet, you will place the sample sheet on a USB or network drive that is accessible to the instrument and browse to it. A new sample sheet can be used to specify a different DRAGEN pipeline than the one that was originally run. If the run was originally set up in standalone mode, meaning that no analysis occurred after sequencing, then a new sample sheet can be imported and used to requeue the analysis. If a new sample sheet is not selected, then the original sample sheet found in the run folder can be used. Now let's discuss the output directory. If the output directory is not changed from the original location, the original analysis results will not be overwritten. Instead, a new subfolder to hold the new requeued output will be created in the output directory. For example, if the original output was your output directory analysis/1/data, then the requeued output will be in your output directory analysis/2/data. Now that we understand how to requeue the analysis locally, let's talk about requeuing on the cloud. The version 1.2.1 BCL Convert app requires that a new sample sheet must first be uploaded into a BraceBase project before use. Then the new sample sheet can be selected and used in the Override Sample Sheet option on the app's configuration page. The DRAGEN NextSeq 2000 workflow cannot be requeued in the cloud. This means if a cloud analysis fails or if FASTQ files need to be regenerated in the cloud, a new sample sheet will need to be used in the BCL Convert app. After running the BCL Convert app, you can then use the FASTQ files as input for the BaseBase DRAGEN apps. When using the apps in BaseBase, please note that the BaseBase DRAGEN apps are not free to run and that a BaseBase subscription is required to run the apps. If a custom genome was used for the initial cloud analysis, then that genome will have to be recreated using the BaseBase DRAGEN Reference Genome Builder app in order to use that custom genome with the BaseBase DRAGEN app. We are once again taking a look at a V2 sample sheet, and I wanted to point out that the FASTQ compression format here is set to DRAGEN. If this is the case, when using the BCL Convert app, the analysis will fail. And this is because, as mentioned earlier, DRAGEN compression to FASTQ.ORA files can only be done locally on the DRAGEN server. The BCL Convert app will always use a gzip compression for FASTQ files. Another thing to note is that the BCL Convert app requires cloud data section to be present in the sample sheet. When creating a sample sheet and instrument run setup, it'll automatically create this cloud data set. The project name column can also be used to assign samples to projects. If the project name column is omitted, then samples will be written to a project named after the run name from the header section of the sample sheet. Here are some of the BaseBase apps that can be used once you have FASTQ files that are generated by the BCL Convert app. Again, please note that to run these apps, it does require a BaseBase subscription and iCredits. Now we are moving on to the second half, where we will discuss what to expect once the NextSeq 2K DRAGEN analysis completes. Let's start with the location of the DRAGEN output files post-analysis. When analysis is performed in the cloud, DRAGEN output files are found within the analysis app session. A link to the analysis app session can be found in the Run Summary page as shown here. The analysis app session can also be accessed from the project associated with the run. And for cloud analysis, the output files will include FASTQs, BAMs for all DRAGEN pipelines. When analysis is performed locally and saved to a local output directory, i.e. a network drive or external drive, the DRAGEN output files are found in the analysis folder within the run folder. When analysis is performed locally and then uploaded to cloud storage, DRAGEN output files are uploaded to a BaseBase project. Clicking on the link to the upload session or app session shown here will take you to the app session within the BaseBase project. Within the app session of the project, you can click the Files tab, and there you will find the DRAGEN output. The output can include variant calling, RNAquant, RNA fusion, and depending on the pipeline run and log file. Please note that for local and hybrid runs, for all workflows, including BCL convert, that FASTQ files will not be uploaded to the cloud, even if cloud storage is used. You will need to use cloud mode if you would like for the FASTQ files to be present in the cloud. So now that we understand where to find our output from the NextSeq 2K and what files are present, let's talk in detail about the DRAGEN pipeline. First, we'll start with the DRAGEN FASTQ generation workflow. As with all of our workflows, DRAGEN FASTQ generation will start with the input of the run folder and sample sheet. Then the BCL convert software will generate the FASTQ files and FASTQ stats as output. For the DRAGEN FASTQ generation workflow, version 3.8.4, there is a no lane splitting option that has been enabled. This changes the behavior of the FASTQ file generation for P3 high output flow cells. Now FASTQ files for P3 runs are consolidated into one FASTQ file per read. So for each sample, you will get one read 1 and one read 2 file. And this is consistent with the behavior of P2 flow cells. And this affects FASTQ files again for the output of all version 3.8.4 DRAGEN workflows for P3 runs. This is the analysis run setup page. This is an in-instrument run setup. And right now, this page is specific to the DRAGEN 3.7.4 workflow for FASTQ generation. We can see that the default value here for barcode mismatches is 1 for BCL convert on the DRAGEN NextSeq 2K. This means that the indexes off of the sequencer can only have a 1 base mismatch for the corresponding index present in the sample sheet to map the read to the sample. This parameter can be changed, and let's touch upon configurable settings. First, we have the adapter trimming settings. If you select an Illumina adapter kit, you will not need to configure this setting. It will be on by default. However, if you use a custom kit, as shown here, then you will need to, in the Create a Run section, is where you would indicate the adapter sequences to trim off of the 3' end of the read. Here, you can also change the barcode mismatch setting from the default of 1 to 0 or 2. This slide shows an example of how to use the override cycles configurable setting to accommodate libraries with UMI. The override cycle setting is similar to use bases mask in BCL to FASTQ. This setting can be used to mask out bases or reads and indexes during demultiplexing. Let's say that the first 7 bases of read 1 and read 2 are UMI, and the 8th base is a linker that we want to ignore. Y in this case means data or the read, N means to ignore the base, index, I is for the index read, and U is for UMI. So in order to complete the first example, we would put in this override cycle setting on the sample sheet, UN, U7, N1, Y143, followed by I8 for the index read, and then U7, N1, Y143. The override cycle setting can be included in the settings section of the sample sheet. Here is the indexmetrics.out.bin binary file. And this is a binary file which contains a summary of the demultiplexing results and is the output from the DRAGEN generate FASTQ workflow. The contents of the indexmetrics.bin file are visualized either in BaseBase's run indexing QC tab or in the sequencing analysis viewer, also known as SAV. For cloud, hybrid, and local runs that use cloud storage, the indexing QC chart in the base run summary page will populate upon the completion of the analysis and the completion of the file upload for hybrid and local runs. For local runs that only use local storage, the indexing QC metrics tab can be viewed in SAV. For local runs, this slide shows SAV and the indexing QC metrics that can be viewed here. To view the indexing metrics in SAV, we must first copy the index metrics out.bin file from the reports folder into the interrupt folder, and then you'll be able to launch SAV by browsing to the run folder to view the indexing metrics. Shown here are really helpful files for troubleshooting demultiplexing issues. The demultiplex stats shows the reads assigned to each sample, and the top 100 unknown barcodes shows index read by the sequencer that were not assigned to a specific sample. These files are also described in detail in the NextSeq 2000 system guide. And lastly, for this section are the DRAGEN FASTQ generation workflow hard-coded options. These are hard-coded on the instrument and cannot be edited from the sample sheet. A commonly asked question is if the use strict mode setting, which is defaulted to false, when true, FASTQ generation fails, and then when there are missing or corrupt BCL files. And when false, FASTQ generation can proceed beyond missing or corrupt BCL files. Although not shown here, this option is set to false, meaning that FASTQ generation can proceed even if there are missing or corrupt BCL files in the DRAGEN FASTQ generation workflow. Now let's move on to the next workflow, which is DRAGEN Germline. This workflow can be used for whole genome sequencing analysis. Shown here is the general pathway of the DRAGEN Germline workflow. It will take in the run folder and perform alignment and variant calling. What's new with this workflow on the NextSeq 2K is the CYP2D6 caller, which has been added to the workflow. The CYP2D6 caller has integrated Cyrus and requires DRAGEN version 3.8.4 and above. CYP2D6 requires a special variant caller because of a wide variety of common structural variants and a similarity to the pseudogene paralog CYP2D7, which makes it difficult to genotype. You can find more details for the CYP2D6 caller in the Illumina DRAGEN BioIT platform user guide. In the analysis settings sections, when creating your sample sheet for the DRAGEN Germline workflow, you will have the option to select the map and align output format. A CRAM is an alignment file similar to a BAM file, and it is a compressed version of that alignment. CRAM files reduce the disk footprint of the alignment data. If none is selected for this setting for the map and align output format setting, no BAM or CRAM file will be saved. All other expected output, variant calling output, alignment, and coverage, and variant calling metrics will still be output and saved. The option to select none is only available when the output location is selected as local versus base-based. These are some of the hard-coded options for the DRAGEN Germline workflow. Not shown here is that the Illumina annotation engine, Nirvana, is not enabled, meaning variants in the VCF and GVCF will not be annotated. I've listed out some of the output files to look for here for the DRAGEN Germline workflow, including the HTML report, the mapping metrics.csv, and the whole genome sequencing metrics.csv file. Next, let's move on to the DRAGEN Enrichment NextSeq 2K workflow. What's new with this workflow is that users can now optionally use a noise baseline file to improve their somatic variant calling. A noise baseline file is used to filter out sequencing or somatic noise and is only supported for somatic variant calling mode. And it also requires DRAGEN version 3.8.4 and above. Custom noise baseline files can be generated using base-based DRAGEN Baseline Builder app or the standalone DRAGEN BioIT platform server. Another thing to note is that for the small variant output, in Germline mode, both the BCF and GVCF file are generated, whereas in somatic variant calling mode, only a VCF file is generated. Shown here is an example of a DRAGEN bed file. A bed file stands for Browser Extendable Data. Variant calling is restricted to the target region specified in the input bed file. The bed file should contain the chromosome, target, and stop and start coordinates, as well as the target region name. The alignment enrichment metrics will be calculated based on the alignment to the specified target regions in the bed file, which is why the bed file is required for DRAGEN enrichment. A schematic baseline file drop-down menu has also been added to Instrument Run Setup. You can either select a standard file or upload a custom noise baseline file. For local and hybrid run modes, users now have the option to save the FASTQ files for their local output when using the Keep FASTQ Files option shown here. The default for this is no. Please note that when using cloud mode, FASTQ files are always saved in BaseSpace. These are some of the hard-coded options for the DRAGEN enrichment workflow. The enable duplicate marking and BAM indexing and sorting are all defaulted to true. Many additional per sample metrics files can be found among the DRAGEN enrichment workflow output. You will get the HTML report, QC coverage region.bed files, CSV files, and the mapping and variant calling metrics. The summary.csv will also be output as well, only when using cloud mode. Now moving on to the DRAGEN RNA workflow. The DRAGEN RNASeq workflow performs alignment and quantification of the RNA reads, gene fusion detection, and optional differential expression analysis. This slide shows the RNA alignment and quantification workflow. The DRAGEN RNA workflow quantifies RNA expression and determines the relative abundance of different RNAs. The DRAGEN Quant module counts and quantifies reads aligned to the genes and transcripts in the reference annotation, and normalizes those counts using TPM, or transcripts per million, mapped reads method. Ribosomal RNA filtering is also a new feature that has been added to the DRAGEN RNASeq workflow and also requires DRAGEN version 3.8.4 and above. This slide shows the fusion detection workflow. Gene fusions are called based on reads mapping to or split between two different reads, two different genes. These split reads are chimeric reads or supplementary alignments in the BAM or CRAM file. The fusion detection module is not looking for predefined fusions, and novel fusions can be detected. rRNA filtering has been added, meaning that RNA reads are marked in the BAM and excluded from the fusion detection analysis. This slide shows the optional differential expression analysis workflow, which also requires version 3.8 and above. The DRAGEN RNASeq workflow performs per flow cell differential expression analysis. And differential expression analysis can be set up to compare gene expression levels between samples in the same run. To compare samples from different runs, you can use the BaseSpace RNA app plus the BaseSpace DRAGEN differential expression app. An RNA annotation file can be added when creating the sample sheet in Instrument Run Setup. This file provides exon boundaries to the aligner and gene locations for quantification and gene fusion detection. When uploading a custom annotation file, the file must be in GTF, also known as general transfer format. If using a preloaded genome, like a human mouse, like the human genome or mouse and rat genomes, if a custom file is not uploaded, the annotation file found within the genome folder on the instrument will be used. Users can also find the gene annotation files from any of the following sources known as, such as RefGene, Ensemble, UCSC, GenCode, ACU, and Vega. What's new is that users also now have the option to enable differential expression analysis during the instrument run setup. Again, differential expression analysis is optional, and the table shown at the bottom of this screen is only used when differential expression analysis is enabled. When running differential expression analysis, you can create the sample groups in Instrument Run Setup. Each group, comparison, or control must contain at least 2 samples. Groups being compared may have mismatched numbers of samples. For example, for the comparison 1, the control group can contain 3 samples, and for comparison 2, the the comparison group can contain 4 samples. The samples are not paired between groups for differential expression analysis. This slide shows an example of a sample sheet with differential expression analysis enabled. And then this is an example also with differential expression analysis disabled. Up to 5 comparison fields can be added to the DRAGEN RNA underscore data section. Use the values control, comparison, and NA to assign samples to the control group, the comparison group, or to exclude samples, respectively. When creating groups, each group, comparison and control, must contain at least 2 samples, and the DRAGEN RNA underscore data section must have the sample ID column listed with the sample IDs required regardless of using differential expression analysis. These are the hard-coded options for DRAGEN RNASeq. You'll notice that the standard settings are the same for DRAGEN Enrichment. Duplicates are reported but are not excluded from downstream analysis. For the DRAGEN RNA workflow, I've listed some of the key output files here. You can expect to find the HTML report, the transcript and gene-level quant results, and the final list of fusions detected for a given sample. For differential expression analysis output, you can also expect to find the control versus comparison.genes file, as well as the differential expression metrics and PNG files for heat maps to visually display the differentially expressed genes. So now I would like to introduce the NextSeq 2000 DRAGEN single-cell RNA new workflow, which requires DRAGEN software version 3.7 and above. The DRAGEN single-cell RNA workflow supports single-cell and gene classification and counts unique reads as UMIs per gene cell. The RNA aligner step requires FASTQ and the gene annotation file as input. Reads are aligned to the reference genome, and for 3.8.4 and above, rRNA filtering filters out the read pairs that align to rRNA sequences. The DRAGEN barcode and UMI extraction step extracts cell barcode and UMI sequences from the FASTQs. Then the merge step will align the reads that are tagged with their corresponding cell barcode and UMI sequences. The tagged alignments are then written to a BAM or CRAM, and the cell gene classification and duplication steps will occur. The cell filtering will filter out all the cell barcodes that likely do not correspond to real cells based on the UMI counts per cell barcode. And finally, it will output the report.html. The complex workflow steps shown here are similar but not identical to those performed by the third-party single-cell RNA software, such as STAR Solo and Cell Ranger. The main output is the cell by gene matrix file, which reports the expression level or UMI count of each gene in each cell. In Instrument Run Setup, you can select the library prep kit that is compatible with your library. The index adapter kit dropdown will display the corresponding adapter kit name under standard kit. An RNA annotation file is required for analysis, and if one is not selected, the annotation file GTF found in the local reference genome directory on the instrument will be used instead. Users will also need to select the configuration type, number that matches the selected library prep kit, and index kit adapter numbers. For example, we selected library prep kit 2 for single cell on the last slide, and the configuration type here is type 2. Cell barcode, UMI, and RNA library type settings will be pre-configured based on the configuration type that is selected, and these fields can be manually configured for custom libraries. You'll notice specifically for the single-cell workflow that regarding duplicates, UMIs are used for deduplication, and the duplicates are not marked in the BAM or CRAM file. Please note again that all options shown on this slide are hard-coded, meaning that they cannot be changed via the sample sheet in any run mode. From the output, you can expect to see the new aggregated report that pulls together all the following metrics for the samples. Files included for each sample will be the matrix MTS, sample barcode TSV, barcode summary TSV, and gene TSV files. Our next new introduced workflow is a DRAGEN DNA Amplicon workflow for the NextSeq 2000. The DRAGEN DNA Amplicon workflow requires DRAGEN version 3.8.4 and above and requires a bed file as input. All the standard bed files available will be in the DNA bed file drop-down menu, and they're compatible with the HG19 reference genome only. From the target regions drop-down, you can either select a preloaded standard bed file or upload and select a custom bed file. It is important to note that for analysis, the bed file must be based on the same genome build that was selected in the reference genome dropdown, for example, HG19 or HG38. These are the hard coding settings for DRAGEN DNA Amplicon. And please note that the enable duplicate marking is full. Some key sample metrics files found among the DRAGEN DNA Amplicon workflow output are the HTML report, the QC coverage region.bed and .csv files, as well as the alignment and variant calling metrics CSV file. Okay, so now that we have gone through all the DRAGEN workflows, we will discuss briefly on how to check for updates to the individual workflow versions. These instructions are intended for online installation of DRAGEN workflows with version 1.3 and above in the control software. Multiple workflows can be selected and downloaded at one time. And after the installation is complete, you will then be navigated back to the DRAGEN screen and can view the updated list of your DRAGEN workflows. First, you'll need to ensure that the NextSeq 2000 is in an idle state. This means that in the control software process management screen, there should be no sequencing present or file copying. On the NextSeq 2000, you'll log onto the Linux operating software as Illumina admin. You'll then update the DRAGEN workflows by going to the DRAGEN settings page, select check online, and then you will select a checkbox for the workflows that you want to update. Please note that the DRAGEN BCL convert must be installed before or with other workflows of the same DRAGEN version. Then you'll click download to start downloading the installers, and after the download is complete, you will then install to start the installation. After the installation is complete, then you will enter, during the installation before it starts, you'll enter the Illumina admin password and then select authenticate. These instructions here are for offline installation of the DRAGEN pipeline with version 1.3 and above of the control software, or offline installation with 1.2 of the control software. The install can take about 20 minutes, and similarly, you will download the DRAGEN workflow installers in tar.gc format from the NextSeq 2000 support site. You will then save these installers to a portable drive or mounted network drive. Note that, again, BCL convert must be downloaded and installed before or with other workflows of the same DRAGEN version. If you save the installers to a portable drive, you can then plug those into the instrument. You will need to ensure that the instrument is still in an idle state, meaning that no process is running. On the NextSeq 1000 and 2000, you'll log in with the Illumina admin password, and then to update the workflows, you will go to the DRAGEN settings page. Under the version, select Browse for New Version to navigate to the installer. Select Install to start the installation, and then log in with your Illumina admin password to authenticate. So please note that only workflows of the same version may be active at the same time. If a newer or older version of a workflow is subsequently installed, only workflows of that same version will be active. We will discuss switching between different software versions in a bit. But first, I wanted to show, since there are multiple versions of the NextSeq 2K control software and the DRAGEN software that are available, for reference, this slide shows which versions of the NextSeq 2K control software are compatible with versions of DRAGEN. So you'll notice on the last two versions that the single cell RNA and Amplicon are compatible with both the NextSeq 2K control software 1.2 and 1.3. So now we'll talk about switching between the different DRAGEN software versions. You can have multiple different versions of your DRAGEN workflows installed on the instrument at the same time. DRAGEN version switching will occur between hybrid and local run setups. When the user selects to set a run by loading a sample sheet or selecting a planned run where a non-active version of DRAGEN is specified, for instance, if the user uploads a sample sheet that specifies version 3.8.4 of the DRAGEN enrichment workflow, when the active DRAGEN version on the instrument is 3.7.4, you will then be prompted to switch to 3.8.4. And then you will, as seen on the screenshot at the left. How version switching works is that you will load a sample sheet, select a planned run where the analysis specifies a version of DRAGEN that is not active on the instrument. The control software will then prompt you to proceed with switching the DRAGEN version. And then while the switching is in progress, you will see a message indicating that the switch can take up to 20 minutes. When the switch completes, the user will then be prompted to the run review screen to switch to the DRAGEN version. The DRAGEN version or workflow will be specified in the sample sheet must already be installed on the instrument. And both Illumina admin and Illumina users have the ability to switch to your desired DRAGEN workflow, as long as that workflow is already on board the instrument. Next, we'll look into the DRAGEN reference genomes available on the instrument. All of these genomes are already converted to DRAGEN hash tables for use on the NextSeq 2K. The following genomes can be downloaded from the DRAGEN support site for use on instrument. The following graph genomes can be downloaded for use on instrument, which are the HG19, HG38, and HS375 graph genomes. It can take about 10 to 15 minutes to import a custom reference genome onto the NextSeq 2K, so you'll need to plan to start your run setup accordingly. So the last thing we want to touch upon today is the secondary analysis output files that get transferred by Universal Copy Service. This section is specific to local and hybrid modes that will have the output files copied to the cloud, local storage, or both. For transfers to the cloud, it will transfer workflow-specific files, such as BCF or differential expression analysis results, sample metrics, the log files, and the reports folder. Files transferred to local storage will be BCF and GBCF files, the sample metrics, log files, and the reports folder. The option to also transfer the FASTQ and BAM files, BAM and CRAM files from alignment will be present. However, for cloud analysis, FASTQs and BAM and CRAM will not be transferred to the cloud. To save space, FASTQ files are automatically deleted from the on-instrument temporary run folder before the data is transferred. To save these FASTQ files, when you're setting up the run and instrument run setup, you'll select yes to keep your FASTQ files, and it will show as keep FASTQ true in the sample sheet. Please note that if FASTQ files are wanted to be present in the cloud, you must use cloud mode and not local or hybrid mode. Okay, so that is all the information we have for today's webinar. I know it was a lot of information, so thank you very much for taking the time to join me today. I hope this session has been very helpful for you.